process with simsMVA. So we covered the spectra mode, the depth profiling, and then we started to look at the imaging mode. So we exported the data and did some PCA. So I won't continue from this point. So now we'll do some NMF of the same imaging data set. So let's go back. Yeah, so here's just a reminder. Yeah, so that's what we got from the PCA. And we were exploring the components showing these features in this adhesive sample. So now, okay, so now we return to the tab where we loaded the data, and let's perform NMF. So if you remember, for NMF, you have to remove the main centering, and because there's this TOF data, we will do Poisson scaling. So that's what it looks like. Also, if you remember, normalization here, so we're normalizing all the maps by the total map. So select it here. That's all we need for NMF. Also, we discuss the possibilities of using binning and smoothing in case your data is noisy or you're too large for the processing. But it's we can live with this side. It's all right. So the NMF button is the same as we have for the depth profiling or the spectra, if you remember. But there's one thing that's important for the imaging, which is something I mentioned in the first seminar, which is the subsampling of pixels. If you remember, I said with SIMS data, many of the neighboring pixels are correlated. They do have very similar signal. So what we can do is we can do PCA or NMF only using some of the pixels, not all the pixels. And then once we find the components or end members, we apply this to the entire data set. When we do this, we save time. It takes much less time, and most of the times the results are the same. The limitations are, let's say you have tiny features that may be missed when you randomly pick pixels, But in this case here, if we do, let's say 10%, it's quite likely that we will get all the chemistry going on here. You can always test. So to illustrate this, I will run first NMF using all the pixels, and then we'll do it again using, let's say 10%, and see the differences. So again, sorry, I'm recording this session. I forgot to start recording. Are we? Okay. Thanks. So yeah, so remember, things we have to set up always is number of end members and number of iterations. And we'll keep the lack of fit calculation to see how good the factorization is. So let's say, you remember we had the kind of the matrix region, and we had something inside and something on the edges of those features. So let's say there are 3 components or 3 end members. And let's try to run this with 500 iterations. So yeah, so that's how long it takes. It's not that long, but if you're exploring things, trying different normalizations or range of variables or things like this, pre-processing, it may in the end it will take very long time if you do it several times. Also, all the data sets are much larger than this. You have many more peaks and many more pixels. And yeah, the more peaks and more pixels, the longer it will take. So yeah, so before we look at so it creates a new tab with the NMF, like it does with PCA, etc. So let's now do 10%, same conditions. No, it was 3, right? This, yeah. Yeah, see, it's much quicker, obviously. If you do on 1%, it will be even quicker. Right, so you go there, it's like we always have the top panel showing here the intensities of the end members. So we covered this already. So there's no negative values now. So each end member will look like a characteristic spectrum from a compound, let's say. And here you have that same window that we were using to explore this course. So before we look at the intensity windows, here in PCA, if you remember, let's go back there. You had the variance per principal component. Yeah. Now in NMF, you have the error of the factorization per iteration. So we saw that for the depth profiling, right? So we can see whether the factorization has converged or not. But also I show here the error of the image. So basically how good the factorization is against all the data sets in a kind of pixel by pixel visualization. And you can see we're not quite factorizing everything. So it's quite likely that we could have added another component, or another end member that would cover these things that are missing. So instead of 3, we would need 4. But it's all right. We can see what we got with 3. But it's important to check both these graphs here to see if your factorization is correct. Similarly to PCA, you have the maps here for each of those. And if you select the bottom one, it will change the y-axis in this scatter plot. And notice that it's non-negative, right? There's nothing less than 0. Again, with PCA, we're picking diverging color maps, but now because we have only things going from 0 to something, it doesn't make sense to pick divergence. Actually, it doesn't look good at all. Some of them will do, but it will not help you with your interpretation. So we can use something like this hot, or like use last time the blues that will look orange, or yeah, brown. So yes, that's the first one, kind of the features. Second one seems to be the matrix and something inside as well, the features. And the third one seems to be, oh yeah, that's why we needed 4 probably, because we had this kind of this blob here. And let's have a look at number 3. Yeah, we need to check the peaks to confirm, but I'm quite sure this was some contaminant, you know. So, and obviously it's got quite strong signal and many pixels in here. So one of the end members was dedicated to this. So like we're talking about having the inner part and the edge of the features, we couldn't factorize into separate these two things because we only have 3. So let's try to do it with 4 then. Let's go back to the imaging panel, and now it was this one. Let's go back to 100% of the pixels, and then now do it with 4, right, instead of 3. Let's see now. Yeah, number 2 now is showing that contaminant. 3, 4. So now if you look at 1 and 4, we managed to separate the edges from the cores of these features. So, you know, this could also be an artifact, but we normalize. So we're trying to make sure this is not a topography artifact. And also for each of your samples, it will depend on how much you know about your material to know whether or not this is correct. But okay, so let's explore this one instead. So yeah, like I said, we have these two maps of each of the end members, and because now we have it non-negative, one tool that's very useful is the overlay. So here is a button called overlay, but we do not click it for PCA because it doesn't help much. But now with NMF, if you click there, it's quite similar to the overview in the depth profile, if you remember from last week. What we get is this window here where each of these drop-down menus will be for a specific end member. So like you can just start by putting all of them. And then you can just change the colors manually or use what's called like a base color map. So this is the color map for the individual maps here. So say like it's the browns. And here you have a set of what's called the discrete color maps where the colors are very different because now we're assigning one color for each end member. Remember from the seminar I was saying we can do this sort of overlays by mapping the intensity into the transparency of each color. So that's what we're doing. So like for example, discrete 1, discrete 2. So all these things will depend on how your features are distributed. And this HSV is good, so as well. The contrast is very good. But you can also do some extra post-processing here, clicking the second drop-down menu here. So you can fix the contrast. So, you know, it will help you to see. So when I have something like this, some substrate things like this, I tend to change it to some sort of grayscale, gray color. Yeah, I think it helps with the contrast. So yeah, so you can also normalize everything by the total. You see how it looks like, or by the maximum of each of the maps. And this is useful when you have shadowing, so we will not help here, but you can change the scale to a square root scale of the intensity. You enhance the noise, but when you have regions with shadows, you also enhance the signal there. But in this case here, the fixed contrast, or even the original one, looks good. So as long as you know what you're doing to your data, it's fine to do this, you know, to show you your results. And here, you at the same time have this overview of the end member, so you can zoom in, and it will do it for the others. So I use this a lot in presentations because it's a quite nice overview of the results. You know, it shows all the peaks, the individual maps, and also an overlay. So it's good for discussions and things like this. You can also do binning to compress, or smoothing like we do with the original data. Okay, so yeah, this is very useful. But remember, if you do it for PCA, it will not look that good because you're going to have, you know, negative values, positive. This will be fine on the right hand side here, but the overlay will not work. Right, so another useful thing is the line scan. You can do it for PCA as well. So if you click there, it will say draw a rectangle and right click to create a line scan. So let's say something like this. Then you right click, you can say create X line scan and number of components. So let's say all 4 of them. And then it just creates it, just showing you the pixel scale here. So it creates one line profile for each of the end members. So you can see how it looks like. So it's just different. In this case here, I think the image is better to see, but sometimes you want to look at interface of material. So you use this signal to identify where the interface is and things like that. So it's similar. I think, you know, in Surface Lab, you also have the line scan tool. So now what we're doing is the line scan after we do NMF or PCA. Let's see. Yeah, you know, the normalization things I was talking about in the overlay, you can also do here before you do anything else. You can do in this main panel here. The auto scaling will only work for PCA because you mean centering again. Yeah, so that's everything about the imaging. So we did PCA and NMF. Hopefully covered most of the useful tools. There are more buttons here, as you can see, but feel free to explore them. But these are the main ones, the most useful ones, I would say. So now let's try to do some exporting and then processing of 3D data sets. And as always, I will just wait to see if there's any questions. Let's say 5-10 seconds.
Quick question, Gustavo. When you were choosing the iterations, you chose 500. What was the reasoning for that?
No reason actually. I guess it's experience from this data set. It's usually from tof-sims data sets, imaging specifically. I always have to do more than 100 from experience, but there's no reasoning, in terms of any mathematical reason. It's just, well, if I don't know anything about it, I would do first, let's say 500, and if it's not converged, I would do more. But then if I know that it converges to 500 and I want to do quick tests, I maybe do less. So it was, yeah, there was no reasoning.
Okay, thanks.
Right, so you remember last week we were looking at the depth profile of this device. Where is it? Yeah, we used the other window. Yeah, so look at this depth profile with, thick layer on top and then 2 thin layers and a substrate. So now let's use the exact same data set, but instead of exporting the data, like let's say this on 1D or only depth profiling data, let's explore all the voxels so that we have a 3D data set. So what we do is, the protocol is the same, you have to create your pick list, select the peaks, and then with this Surface Lab 7, you just go to the other software modules like imaging, depth profiling. The peaks are already there. So yes, create a pick list. And to export 3D, now we have to come to the imaging software. So quite similarly to what we did with the images data set with the adhesive, just go File, Export, and then again select all the peaks you want to export. Now the difference is you have to export scan resolved images. So first you can select a range of scans, or if you want everything, you just do everything. And then you go scan resolved images. What you can do is you can combine. So, this is quite a large data set, and it's quite likely that if you get the map for each level, it will be very noisy if you're doing not long acquisition time per sputter cycle, So you can join, let's say 10. So you can have, instead of 8,000 points or 8,000 images, you're going to have 800 something. So, but if you want to export 3D, you have to, do scan resolved. And then again, you have 2 options. We can do the binary, the viv6, and it works in the same way. You select a folder, and now whichever file name you give here, it will be the base file name, because it creates one file, viv6 file for each level when you do the binary. So it will, if you have 8,000 scans, it will create 8,000 files. So it's for each scan or depth level, because it's a depth profile, right? So I will not do it now because it takes a while to export, but that's how we do. The other option is similar to the images, is to use the ASCII. So now it will create a file with the X, Y, Z coordinates and intensity for each of the ions. So the difference is it will not create one file for each level, but it will create one file for each ion you have. So sometimes you don't have as many ions as levels, so it may be advantageous to do this instead. And also, I recently found out is in this newest version, at least the version I'm using, there's a problem in Surface Lab when you export using 3D data using BIF6. For some reason, the first levels are not, they're kind of merging more levels into them. I don't know what's happening. It's like you have, the first 100 levels together in the first one. So I think for now, it's if you have the same bug, maybe we should all use the ASCII for the 3D. Unless you exclude the first level, the first few levels after you load. So yes, again, I will not do it now, but let me show you what it looks like. So with the text files, you have one for each, one file for each ion containing data for all the levels, all the scans. With the BIF6, you have one for each scan. So here I was merging several levels. I was merging every 100 levels. So I end up with 82 files only. So once you export it, you go to simsMVA and create a new tab. Now you select 3D. Give it a name. And then you click that load button. Load file. And these are the two options. So let's say do BIF6. Let's find it. So if you select all the files and go open, it will provide you with this option which to allow you to compress it even more if you want. You can merge every 2 levels now, so you end up with 40 levels instead. And you can also compress each image. If you have 256 by 256 pixels in each level, you can compress it to 128 by 128 and etc. before you load, in order to save memory. Because when you do 3D, you find that it takes up lots of your memory in simsMVA. The way I do it, I load it up in the RAM, so you have to be careful. So always check your RAM if you're dealing with a really large data set. So yeah, make sure you're using a good computer. So yeah, and then it loads all the levels. Similar to the image, it asks whether you want to crop it. If you say no, it will just load everything. Yeah, so that's what it looks like. So this profile is a bit strange because of that bug I said. So let's now load the text files instead. So I selected the ASCII files, and I go find the files, and I think I mentioned it last week. I still have to fix it. We have to skip the total because it's crashing when we try to load it. I'll fix that. One thing you notice is that those text files are much larger in general than the BIF6. So the binary is always good because it doesn't take up much of your hard drive space. So unfortunately, now we have this bug. So maybe for now we have to do this. Yeah, it takes a bit longer as well. think I have it loaded in another window, like in those baking shows, the cake's already ready, right? So yeah, I have it here. So let's stop that loading. Okay. So I actually fixed the problem with the 3D. So we have a total of the list here to normalize to if we need. So basically, yeah, so that's what it looks like once you load it in the 3D. So it's quite similar to the imaging. You have these menus to see, to select specific ion, then normalization, binning, smoothing. But because it's 3D, it takes a bit longer to do each processing. So you saw that just now, because it had this level 3 of smoothing. It will do it for all the, it's kind of a function that takes into account pixels and also levels. So it takes a few seconds to process. So yeah, be careful, if it's too large, you may have to wait for each of those things you do. I may in the future add a button to process so that, if it's too large, you don't have to wait every time. So yeah, it's already in the post scaling here in this case. And now, even more important than images, the subsampling is very, very useful. Not only because it will save you time, but imagine it is not the largest data set, but imagine you have thousands of peaks, or has more than 500 peaks and several levels with high resolution, and you want to keep this high resolution. First, if you had the memory to load all this and process, it will take forever. It may take, let's say, half an hour to process. Secondly, you will not necessarily have this memory. So if you process just a few of the voxels, you can fit into your computer memory, and then you get the results afterwards. Just remember, I forgot to show the results with the 10% in the imaging. We can go back there later. Sorry, I forgot to show. But so let's continue here first, otherwise we just lose track. So let's see what's. Yeah, so let's try to run NMF in this 3D data set. I will normalize by total. Okay, so it works in the same way. You click, and that's exactly the same window. So because there's, remember we joined up hundreds of levels, every 100 levels. So those very thin layers, they quite likely became just one now. It's compressed down. So we can try to do it with 3, just to illustrate what's going on. And then again, arbitrary. I don't remember how long it would take for this one with 5% of pixels. Let's say 200, just to see. Run. Yeah, that's not bad. You can wait this one.
Gus, could you give us a refresher why when we're doing NMF, we do not need to do any mean centering?
It's because when you mean center, you bring values to negative numbers, because you'll be around 0. The new 0 is your average. So things under 0 are going to be lower than your average. And NMF is non-negative. Your results are all non-negative. So when you do NMF, of any data set containing negative values, all these negative values will become 0 and you lose this information. So we can't mean center. And there's no reasoning for doing mean centering in NMF as well.
Understood. Thank you.
Okay. So it then creates a new tab. We've got quite a few there, but it always goes to the end. And what you get here for this panel is only the lack of fit. And the other two are very similar. This one is actually the same. You must be familiar with this now. The end members. And here we don't have those scatter plots anymore because now we have a kind of level view and the 3D view of the data set. So you can select each of the end members. So looks like you found these 3 layers. And this drop down menu here is the levels. So let's say you go to level 29 and then it will show the image here for level 29, and it will also show its lights of level 29 in the 3D. But you can also do the overlay. In this case, per level, there's not much contrast. So let's go to that overlay. It's very similar to the imaging that we just did. But each layer seems to be quite homogeneous, right? So you won't see much. But then if we go down, let's say to closer to the interface, yeah. So here, around here, when things are changing, you can overlay. Let's use the same colors we had before. Then you can see level by level things changing. You know, that's turning from, it's going from the gray into the red end member. And before you had the green. This film button will create, not planning to cover this today, but just to let you know, it will create a video going level by level at specific frame rate that you set. So sometimes it's useful as well for presentations, because if you show kind of a video of your data showing, especially in this case here when you reach this level. So, you know, I'm pressing arrow down. I click here and pressing arrow down on my keyboard, and you can see it changing. Because now we have this information. Right? Before it was all compressed into a depth profile, but you can see here there's some XY information. It's like there's not the change is not sharp. You know, the reason behind this could be because of differential sputtering, or even because the way the material is created or was printed is not homogeneous, and the thickness is actually not homogeneous. But we can also see an overlay in 3D. So if you click 3D overlay, you ask whether you want to do Z correction. In this case, we don't. So if you remember, it was to do with having topographical features on the top surface. So in this case, we assume it's fairly flat. So this is a new version of the overlay. So it's under development, but I thought it would be worth showing you. So basically, if we focus on this panel here that says colors, you have each of the end members here for the colors, and then you can change the colors like we did for the 2D overlay just now. So, you know, you just change the colors. So these things are to do with capping the volume that we will render. We just enable everything. And this is to compress and make things quicker. I will show you in a second. So once you It's a bit confusing now, so I will just show. Hopefully, in the future will be less confusing. So here you have three options: isosurfaces, scatter plots, or the fast scatter plots. So let's start with the isosurfaces. I don't know if you remember from the seminar, I showed some examples with isosurfaces and scatter plots. So when you click auto plot, it will create it. So here you have this, it's called an individual view with each of those, and here you have this combined view. What you can do, so yeah, it's already showing the volume, and as we said, it's quite homogeneous until it starts to reach the interface. You can normalize, the fixed contrast, all these normalization options I mentioned before. So if to normalize by total, you construct it with the normalized data. And you saw it does sort of layer by layer. That's because those isosurfaces are based on these two values here. So again, it's not obvious because there's no labels yet, but you go from 30% to 90% of the intensities for the red, and the same for the other. So you can tweak this. If you only want to show from 80 to 90% of the gray, you change here, and then it will do less isosurfaces. So it's a combination of isosurfaces. It's something that you couldn't do it with the surface lab. So to me, this is very useful. So yeah, so it really depends on what you want to discuss and want to show, because in a way you just rendering things that you want to. So what I try to do in a case like this is I try to keep it consistent. I give the freedom here for the tweaking, but I'm keeping the ranges the same so that I can trust that these are layers. Another thing you can do, I've now removed the auto plot so that I can change things. Otherwise, it will be plotting every time I change. It's the transparency. So it's the third one here. So let's say 0.6% transparency for the gray, and yeah, so 0.6 for the green, 0.5 for the gray. Let's see how it looks like. Yeah, so then you can kind of see through this rendered volume as well. And the final thing is the compression. If you use the original resolution we had, it will take longer. Remember what we're doing here, we're making this overlay of three things, but each of those three things, they carry lots of information about the chemistry, right? Because they are the end members. So if you go to the intensities, they represent a series of peaks that are correlated. So this is quite powerful. Yeah, you can see the larger it is, the longer it takes. It depends on your graphics board as well. Yeah. Let's go back to this compressed version. And here you have the aspect ratio slider. Will probably not be here once this is finalized, but it can be useful to change it because a more realistic view of this. Obviously, you may not be able to show what you want with a realistic view, but this is a very, if we're doing like, I think it's 50 by 50 microns, the depth of this is few microns only. So this is a more realistic aspect ratio of your data. But then you may not be able to show what's going on. But yeah, you can set this here. Those sliders here, remove the transparency. These sliders are for cropping the data. So then you look inside. This one, yeah, because again, each layer is quite homogeneous. You don't see when you cut inside. You don't see, you don't notice any difference. And there's this create video button where you can create a video that will show different slices, like I had in my presentation. So you can slide through all your X, Y, and Z layers. So to explore the volume. So again, it's useful for presentations. So the other two are the scatter and the fast scatter. So let's look at, they're both similar. So the scatter is basically, now we're not rendering another surface, we're just plotting the points based on those same criteria. And you can change the size of the marker. So you can see these are just points being plotted in a 3D space. So one, let's go back to the entire volume. Yeah, so I increase the size. Yeah, in this case, it becomes quite similar. The rendering will be nice because it shows response to some light, some fake light in here. And these are just flat points. And you can also change the marker type and these things. Yeah, like X's. X's are too small, I think. So yeah, that's how you explore a 3D view of your NMF results. Again, this sort of overlay is good for non-negative stuff. So NMF for PCA will not be able to do this. There's also this 3D view where you don't necessarily overlay, but you explore them with these 3D renders. But it's very similar. Well, I did not mention. Yeah, you can do all the normalization things we spoke before. And that's it. Yes. And again, we're doing NMF where we could have done PCA, and this will look very similar. But now instead you would have had this from negative to positive. What's the time? Yeah. Okay. If we have time, we do PCA later, but it will look very similar. So what I wanted to do now is show something else with the 3D, which is the conversion. So once you load it, what you can do is transform it. If you click this button here, it's a convert to 2D and also 1D actually. So it pops up this window here where you can change it from a 3D to a 2D or a depth profiling dataset inside simsMVA. So let's use the XZ planes. So we get a side view. And then I can just crop, can get like just a thin slice of the sample. For example, if I do this, we will cut it. Yeah, thinner slice. In this case, it will not make much difference again because the layers are homogeneous. So once you're happy with this, and this is your new map for the 10, I am at 10, you go accept and create tab. And then it creates an imaging dataset tab. And you can do everything we already discussed, we can post on scale, mean center, normalized by total. I think there will be an artifact in the bottom. Oh, no, it doesn't. Yeah, you can see at the bottom here this weird pictures because it's sort of divided by 0. So you can maybe do some smoothing to compensate for this. Just as an example, or you could have cropped it before, excluded those bottom layers where the intensity is very low. But then you can, I click PCA button, you can do PCA of this new transform data set. So just another way of exploring, you go from a 3D to a now compressed 2D version of it. And then you can do PCA of this cross section. Because remember, from Surface Lab, you can export maps, XY maps only. This is an XZ map, The vertical here is your Z, is your depth. And there's no way to export such a map from Surface Lab. So what you have to do is export this 3D and do this transformation thing I just showed. And you can see how useful this can be, because now you can do PCA of this cross section. It's essentially a cross section, isn't it? So, and then you get your principal components of this cross section. So here, yeah, showing the layers and etc. Like we discussed last week, you can explore the chemistry looking at the Z view. Or you can, well, in this case, you can do it from Surface Lab, but I will just show you anyway. Or you can click the same button. And then convert to depth profile. And then you have to get the X, only anything with the Z, right? Because it needs the depth information. And then it creates, you go accept and create tab. It creates a depth profiling tab, right? It'll be quite similar to what we had last week because it's the same data set. And yeah, you do, and just as doing very quickly as an example, NMF. So you can see that from the 3D, I compressed it down to just a depth profile data set. And I can do NMF, for example, and then find layer structures, etc., etc. So you can see here foreign members showing the layer structure and their intensities here in the top panel. So this can be very useful if you deal with 3D things or samples with real 3D information. Okay, so before I finish, I wanted to, because last week I was doing the spectra, and because the carboxylic acids were not too different, were too different from each other, we couldn't see really well how to see the groups and things like this. So we just used this built-in example here with in simsMVA, one of the spectra. So we got examples spectra. There's one for wood. We use this one to show very quickly some of the things I wanted to show last time. But because I've just finished this 3D, I'll wait before I do it. 10 seconds, just to see if there's any question. So, yeah, very quickly. This is when you load the example, it's already, the groups are already set. So what we have here is several spectra from samples of filter paper, reference cellulose, reference lignin, and wood. And these are the main components of wood. So I would do standard PCA, normalize, mean center, Poisson scale, and PCA. So what I wanted to show is when you do like PC1 against PC2, I think we have PDMS one, this one here. That's PC1 against PC3, PC2 against PC3. Yeah, so basically what I wanted to show is that when you do this, you tend to get these things grouped in clusters, right? So here, because I said these two purple groups are wood, it considers it as a distribution of a single sort of sample, and it creates this huge confidence interval. And clearly these are two different groups. I know they are, you know, because we took it from different regions of the wood. So it's quite nice to see there's differences in chemistry, but in this case here, they're all called wood, so you know, they will not group together for this specific principal component. But that's what I wanted to show, you know, you can see your samples clustered together in these scatter plots and classify it really well. You know, let's say now I have a new sample and I want to see which part of the wood it was from. If I measure it and I add to this data set, I may see where it falls. You know, if it's in this quadrant here or here at the bottom, I will know how similar it is to my, let's say this is a reference database, and also how much lignin or cellulose is there based on some of the other components. I think the first one, yeah, the first one groups lignin against filter paper and cellulose. So that's what I wanted to show. And I remember last week it was quite difficult to say these things because the samples were really well grouped and it was very obvious that they were different. So now you can see sometimes, especially I know those of you dealing with biological samples, it will never group as well as, you know, standard acids. So wood is one example, but there are other cases which is that are even worse. So you get kind of an overlap between groups. And when you do principal component PCA and then scatter plots between principal components, it may help you to see what's going on. Okay, I managed to finish in time. Yeah, that's everything pretty much. Let me just check.